Thanks, Claudia. So the problem with having me moderate is that I'm not going to stick to any script. So forget everything that you prepared, delete it. Let's be spontaneous. I know it's challenging for our industry colleagues to be spontaneous, but let's hope we can have a free-flowing, good discussion. So the interesting mix here is that some of you already have a product on TB, and it's already out there. Ellen, David, and Chandi to some extent. Some of you are playing on the fringes of TB and gently dancing with TB to check whether you want to really go steady or not. And others are like, yeah, I couldn't care less about TB, right? So let's get these different perspectives. Let's start with the known entities. So Ellen, this is a question for you, if you can take the mic. So you have this fabulous experience of running 17 million cartridges, right? So, and you have that little box that's making its rounds.
It should keep moving. Please send it on.
Right. So firstly, I want to congratulate you because you went ahead and you did not sit back on your laurels or your success, and you innovated to develop this product. What was the thinking behind pushing the boundaries and continuing to innovate versus just allowing your existing product to roll as it is? And secondly, what aspects of that 17 million cartridges worth of 5-year experience are you going to bring to bear to roll out this product more successfully than the previous version?
One aspect, I think, is that a point-of-care instrument like this also has a lot of potential for marketability in Northern Europe and United States. We're thinking of Walmart and Walgreens and physician offices and places like that. And so there is a profit motive, of course, to have a simple single test that could be used for multiple types of tests even in a patient's home. I can see an HIV patient doing their viral load monitoring at home. It's cheap enough that they could buy it themselves. Secondly, one of the issues that we were challenged with was maintaining the instrument, the GX4s, which are the primary instrument in the developing world, and the challenges of electrical outages and spikes and getting dust into the optics. And this thing has been made very much more rugged, solid state, no moving parts practically. And the third part that we wanted to innovate was to be able to have real-time data transfer to cloud-based data aggregation systems, which, for example, in the United States, BARDA has been sponsoring a trial of a similar data repository for influenza. So instead of getting syndromic results or real data 2 weeks later, every time a gene expert tests for flu in an emergency department around the US, it immediately goes to the CDC and they can have real-time data aggregation of where flu is going and which flu it is. Your explanation for continuing to innovate is that it's just not a TB innovation. You see it as a broad-based technology that's going to get you a bigger share of the market globally, US, Europe, and go to a lower level of healthcare that currently your current products would not reach you. And then in terms of the lessons learned from the 17 million cartridges used is that you've incorporated those lessons into the design that is more rugged, temperature, power, and so on and so forth. And you've also tweaked the sensitivity of the TB assay. Can you tell us a little bit more about how much you expect that to be and how different is it from your current cartridge? Yeah, we are hoping the current colony forming units per mil lower limit of detection is around 10. And that should be enough to replace cultures. So we're hoping that a patient who has a negative gene expert result will not go on to culture, and only those who are positive would go on to secondary confirmatory tests. So that's the first hope. And the way it's being done is by using those 2 insertion sequences, which are multi-copy, doing a first round PCR to see if those are present, which enables that lower sensitivity, that lower limit of detection. And then to improve the specificity of rifampin resistance, one of our problems with the G4 version was that we have a lot of false positive rifampin resistance in low organism load samples, partly because the cycle thresholds fall off the right side and issues with the algorithm. So now we have 4 overlapping target primer probe areas using the molecular beacon, and instead of just looking for amplification and delayed amplification if there's a mutation or deletion, now we're using melt curve analysis. So we had to develop a whole new computer software program to analyze melt curve, and that way we can not only look at anything different from wild type, but there are certain nonsense mutations or non-rifampin resistance leading mutations which we can identify, and we won't call those as rifampin resistance because we can tell by the melt curve that that's what we're seeing.
And can you tell us anything about the pricing of the cartridge and the unit?
My understanding about, I know for sure the cartridge is going to be the exact same price as our current cartridge, which I think is $9.95 for high burden developing countries that are on the list. And that thing is less than $3,000. I'm not sure 100% exactly. It's like $28.95 or something like that. But I do believe we're going to have to sell it along with a maintenance contract that goes with C360 enablement. And so there will be a 5-year required contract that goes with it that won't be onerous, my understanding, but just to make sure that it continues to operate and that we can monitor.
Good. And one last question for now is that the already thousands of GX4 machines that are running all over the world, you will continue to supply cartridges for them. They will continue to run until the machines die on their own accord.
Yes, and the Ultra test will run on them, as will the HIV, HCV, HPV, et cetera. Everything will run on those, and we will continue to support those until they die. Right. We hope they won't, but.
Thank you. David, so yours was one of the first molecular tests that was WHO approved for INH and RIF testing. And it's a wonderful product, your genotype array of products. Now you have the fluorotype array of products. Would you have both on the market, or do you see fluorotype essentially replacing the existing first-line LPA?
For the time being, we see both tests on the market. We will not stop producing the line probe assays. Well, what I see from the market is that there is a request for an easier readout of an assay, so I guess there is a fair chance that the fluorotype MTBDR has a chance to replace the line probe assays. But we will not force the customers by withdrawing the line probe assays from the market.
Understood. So again, like Cepheid, you've continued to innovate, which is very impressive. So you started off with your version 1, then you had version 2, then you had your second line LPA, and now you have this fluorotype range of products. What inspires you to continuing to innovate in the TB space? And what is your plan for the new drug regimens and the new drugs that are on the market for thinking beyond INH and RIF to a world where we will have new regimens in 5 years, say? What products do you expect to have in place when those regimens come along?
So, well, what inspires us is that we have a lot of knowledge in the field of mycobacteria testing. So it was relatively logical that we are stepping into the development of an assay when we acquired the rights for a new technology platform. So we just went into the field where we have a very good knowledge, and this is TB testing. And the late PCR technology gave us an interesting platform to find an alternative to those line probe assays. So, well, where can we go? In theory, it is possible also to develop an XDR assay. So the next assay, which is coming for sure, is an NTM assay within this year, which is working from direct samples, or which also can be used out of cultures. So, well, the story about that XDR assay is something what is relatively complicated. It's complex. Yeah? Well, it's hard to go to the shareholders meeting and say, okay, I need 15, 16, or even more $1 million for the development of an XDR assay, whilst there are only 500,000 cases in the world annually, maybe 600,000. Maybe it is increasing, but it's increasing not that fast, luckily, that there is a business case. So, well, we made a proof of principle. Well, you will also hear a presentation from Barry, and also Rob, I think, will talk about it. So there is the potential on the technology that we can do XDR, including PZA testing, with light PCR and light on, light off probes. Well, the question is, who is going to pay it? So we are looking at the moment for certain corporations just to justify the business case.
So your anxiety about going beyond INH and RIF into XDR or the new drugs is just simply the R&D costs that you need to put in, and you think that the market for DST is much more limited than a case detection test?
For sure, the market is more limited. It is more the clinical validation what is a pain in the neck. This costs more and more with every month. This will become more expensive. So regulatory affairs is a case, well, the validation of such an assay until we can go into regulatory affairs is expensive.
And so what is your policy plan or pathway to a WHO endorsement for your fluorotype assay?
With the fluorotype assay, we will go the way. We are at the moment building up alliances to go through the WHO endorsement.
Got it. I will open up to the audience once I'm done with the first round of questions. So, Chuck, you have this is for Chuck. So you are one of those companies that I've seen TB on your website for a long time, and I'm still waiting for your assay. Can you tell us what has been your biggest struggles in moving forwards with your TB product, and what help would you like from the TB community, so to speak?
Yeah, I get that question a lot. What's up? When are you going to deliver something? And I'm not going to say money. Okay, so maybe we're a case study in what not to do, because we have been hyper vigilant about TB since the day we founded the company. To, I think, our detriment, to tell you the truth, that I think one of your questions is if you go in and you try to raise money from an institutional investor, what's going to be your pitch to get them to invest in this? And I have lived that world for over 10 years, 14 years now, and we can't seem to get the pitch through to the institutional investors. So I'll give that same pitch I gave with a 5-minute elevator pitch, and then you finish up and you say, well, our assay, our primary assay, we're all about global health, we're about TB, that's it. And they look at you and they go, well, what about NIPD or cancer? No, I'm here to talk about TB. And that's where it continually falls apart, because they will come in and do their due diligence and try to verify, okay, what's it going to cost for what you were saying, regulatory affairs, quality system, and then they come in and they want to inspect your manufacturing. Say, well, we're seeing out there that this is 988. You're telling me that you can make your device for $3 to hit the margins that we're looking for? And no.
And typically, what margins are they looking for?
Well, it's a growth curve, of course, as you launch, but you typically want to get to 70% margins, at least, or 50%, or, get up there. But that really pushes it down, and that's where it's fallen apart for us. So we had to go the slow route, and we've done it with angel investors, and we've done it through public funding, and that's kind of the reason it's just kind of been a bootstrapping case for us, but we're sticking it out.
So how can we help you? What is it that you're looking for? You said you don't want
Well,
money.
Well, I've asked.
You're changing your mind?
I have asked.
Because Jim has already left
asked
once you said
this-
you don't want money, so.
I have asked this community many, many times, and I kind of came in to this saying I don't want to give the talk because I've written proposals, the Gates proposals, fine proposals, and you can't even get plugged into that. So we kind of said we're just going to go around this and try to do it. But what I've come to understand is, as somebody mentioned in one of their talks, that you got to do it with this group here. So here we are, trying it again.
Thanks for coming.
But this does, I should be in the lab right now, working right now. But to tell you the truth, what we really need is, I think, my one solution I can offer the stakeholders is, in your screening process, try to identify the companies that are truly working on your behalf, which is for TB, and using their money, not trying to use your money to further some other agenda, but using their money to solve this difficult problem. And if we don't make the cut for the funding, whatever the funding line is to get plugged into these programs, I think you should still identify in your screening process the companies that are really earnestly working their butts off to get there, and provide at least some connectivity so that you can feel like you're in the system, a mentor, if you will, that is linked to the company that will point you. I'm quickly told everything's out there for you to find. Let's go to your website, go to this website, but it's a very difficult navigation route, and if we just had somebody who's in the know,
Okay, some kind
guide
of
us.
an honest broker
Yeah.
who will mentor you and connect you to different groups.
to
and
navigate this
pathway.
this journey.
Okay, so I'm not necessarily a business expert, but it seems to me, learning from David and Ellen and Safiyed and Hein, that it may be safer to have products that are, in other words, instead of focusing on TB, which has challenges, should you have products like cancer or whatever, which brings you the kind of a revenue that you need and allows you to take a risk or chance with TB? Because all of them have other products. They're not solely dependent on their TB assay, and therefore they're able to put money down and take a chance. Well, if you were starting off with just TB, it seems like a bigger mountain to climb without having your risks balanced with more safer products.
That would be great if you put that right up in bold writing in every document that's posted out there. But, all we see is these growth curves from Safiyed, and everybody's like, oh, let's do it. So,
Do you want to respond
yeah.
to that, Ellen? Go ahead.
I just want to say one thing. It makes me sad to say this, and Michelle Schoonmaker probably will kill me, but our company celebrated 20 years in business this year, and we are not profitable. So just... We have never been profitable.
These guys are really starting to loosen up, aren't they? Okay, good. David, what
Well,
more
we are
can you
30
tell
years
us?
in business, and we are profitable every year.
Okay, now
So, well,
continuing
it is,
this.
it is really the fact that we have also other products, and that we have an interesting mix of products, that we are also taking up chances when it's going around trading certain products. So, well, you have really to see where you get your funds, and then, well, you can afford also a TB product, and well, those products which are longer in the market, like the genotype MTBDR, the line probe assay, they start to become profitable at a certain point. Okay, until a certain point you have to carry them, but at a certain point there is profitability in. Okay, we reinvested that. but not with the pure target in mind to have an alternative for the line probe assay. We are looking for a broad spectrum of other assays on the technology platform of lights on, lights off probes.
So,
And there are coming
that's
interesting products.
great advice. A TB-only platform or technology is going to be challenging, no matter who it is, how good it is. If it's a single disease, especially TB, given that it's what it is, it's a disease of the poor, and the money is available in TB is not even the same order of scale as even HIV, for example. The other suggestion, Chuck, would be just the true prep alone. Given how we are struggling with many molecular downstream tests with the sample prep is the biggest headache, could the prep device alone be sold at a modest cost so that the back end can be used with any other platform? Would that be an option? That's something you're thinking of.
That's certainly why I highlighted it in my talk, is that we're starting to hear from the user community that alone by itself is lysing more effectively, concentrating more effectively, and provides, Michael's the expert in here, but you really have to provide the right quality of nucleic acid for next-gen sequencing, and from what we're hearing is what comes out of the TrueTip is not beaten up and brutalized as much as some of the other sample prep technologies. So we're definitely looking into it.
Perfect. Chandy, over to you. So you have a product on the Indian market, and you are also selling it in the Indian private sector. But obviously, India being the ground zero for global TB, you would think that an Indian product, first indigenous NAT in India, correct? Kudos. That's quite an accomplishment, given like many other products in India. India is great at generics, but India is just starting to innovate, right? So this is a fabulous example of a true Indian innovation, and you have a product on the market. Now, the Indian government genuinely, at least this particular government we have, genuinely loves Make in India as a catchphrase, right? So the Indian Prime Minister is really keen that things made in India should be promoted, and therefore you should be first on their list, and they are, and publicly they've said so, and it's all over the media that they care about this product because they see it as an Indian innovation, and they would like to have an Indian innovation used for TB patients. Given all of the good things that are stars that are aligned, why is it that it's still not validated and fed into a local policy in India? What are the biggest barriers for you to get the Indian government to take the product seriously, put it through the tests, and come up with a sensible, coherent policy?
I think they're taking it very seriously, and that is why.
Can you speak up? Is the mic on? Yeah. Okay. Just speak.
Yeah. I think the government is taking us very seriously, as you correctly said, and the studies have been on for a period of time, and it has taken significant time for us to reach this stage, but we are, I think, at the end of the entire cycle now, and we are hoping that given the focus for Make in India and the focus for India to create more solutions to address its own problems, I believe that we have a role to play in that.
So at this point in time, are you more interested in a global WHO sort of a policy, or is your first, at least you want to get India to have a policy in place for your product? What do you care about?
See, when we designed this product many years ago, when we thought about this product, we thought about not just India, but maybe 70, 80 countries around the globe that are that have similar problems. So we believe that if we defined a product for India and created our own TPP for that matter, it would be relevant for the rest of the world. We still believe so. We think that we have to have the product out in India, getting out to more people. We are already in the private sector, but you know more about the Indian private sector. You've shared horror stories about this all the time. And obviously it is going to take time to scale. The test and treat paradigm is just kind of evolving. And once that happens, I think the private sector will sit up and do whatever they have to do. But the government is essentially going to play a very key role in seeing that companies like us have the incentive to use our own funds, struggle for 15, 20 years, create a product that can go to market, and address local problems. And we are hoping that that will be the case.
I'm actually thrilled that the four-site study is ongoing with your product, and I think part of the delay was you wanted to automate your sample prep. That led to some innovation, and then you had to have your Rift chip ready, which is what they wanted. So that's partly the one taking the time. So let's hope that 2016 is when they will get their act together and come up with some kind of a policy assessment of your product. Again, like in the case of Cepheid and Hein, you are not a TB alone platform, right? Can you tell us, given that India has every single infectious disease known to mankind except Ebola perhaps, what's your plan for chikungunya and dengue and all sorts of strange pathogens that are running around India in plenty?
We don't even test for TB. You think we test for chikungunya?
So all fevers get all antibiotics straight off the bat. No need for a test.
Having said that, I should say something interesting. See, we, when we started developing, was a development company. We were naive bunch of scientists, entrepreneurs trying to create technology. And then we teamed up with Sriram, who is in the room. And the first thing he told us is that you should do TB on this platform. And he is a very successful entrepreneur.
Absolutely.
And it just goes to show that, okay, there is a reason to do TB, and there is also economic return that TB can give, provided, of course, everything falls in place.
So that's a very nice thing that you said. So just so that others know in the room, this was a small startup company, big tech, and they soon realized that they will just not be able to manufacture, certainly not at scale. And they tied up with Tulip, which is a huge group of test manufacturers in India. I mean, malaria RDTs, Tulip is a world leader. So I think that marriage was a very interesting and a very important step to move forward. And both of you are Indian companies, and therefore it made a lot of sense to have that partnership. Another important question for you, Chandi, is when people think that it's an indigenous Indian product, they immediately expect the cost of goods to be much lower than Cepheid or Hein or Roche or Abbott, right? Now, that's because I think it's driven by the Indian drugs being significantly cheaper than any drug anywhere else on the planet, right? So do you think it's a fair expectation that your cost of goods will be significantly lower, or do you really see that as probably an unfair characterization that your costs of goods actually significantly higher than what the expectation is?
I think that is a loaded question.
I warned you, Chandi.
See, we are an aspiring manufacturing economy. We are not a manufacturing economy at this point. So given that fact, everything in India is going to be as expensive, if not more, than the rest of the world. So make in India is not necessarily make it cheap as of today.
So
that may change
is
in
the
the
Indian
future.
government aware of this? Because for them, make in India is not only a nationalistic statement, but also that it will be cheaper, like the Indian vaccines. Indian vaccines are the cheapest in the world, right? So that's what allows India to scale up rotavirus today or hepatitis in the past. So how do you match those make in India with the expectation about pricing?
Give us the volumes that India can give, and then we can definitely make it as cheap as you would expect.
I think that's where probably the magic lies, that India absolutely guarantees volume for everything. Okay, so let's move on to Joshua. So again, you are a company like Akoni, kind of in the fringes of TB, looking at TB, has some interest in TB. What has been your rate-limiting step in actually launching a product and going through the validation and policy process? What is keeping you up at night?
I mean, I think it's the difference is we're a company of plenty, and then when you have plenty, you have lots of opportunities to pursue. So, a uniqueness that we have is a lot of times you'll have a company that's an engineering firm, but they have no idea how to make assays, right? Or you have somebody that's really good at doing bioinformatics, but they have no idea how to get access to an instrument, right? We have all the above, right? We have the sample prep, we have the instruments, we have the components. But as a company, I come from the legacy kind of applied biosystems business, right? Which is the Sanger sequencing, next-gen sequencing, qPCR technologies that were there. And the company's strategy was really, we are going to enable everybody in this room, right? If you need an instrument, great, we're going to enable you with an instrument. You need reagents to go with your manufacturing on companion diagnostics or whatever it's going to be, we will be that supplier. But what the company never actually did is, and it's taken them some time, is I said, why don't we actually just build the content? Right? Why don't we actually take the time? If anybody in the marketplace could actually come in and actually build content, because we have a $100 million bioinformatics investment, right? We make assays really well. We have manufacturing facilities all around the world. We can service instruments from a global footprint. But people are like, wait, well, how would we do that? Why would we do that, you know? And you can show them, it's kind of setting the business case of enlightening people of like, it's been there all along. You know, it's kind of, I always like to give the example of it's like going into like a Costco, right? And sometimes you tell people, hey, you know, you come home from dinner, it's been a long, or you come home from work, it's been a long, you know, kind of night. You say, I want to eat dinner. And somebody's like, there's a Costco over there, right? Or for those that don't know what a Costco is, it's a big grocery store, right? And it's got all the stuff under the sun. And you're like, but I'm hungry. I've had a long day of work. You know, I want to give you my credit card and I just want to buy a meal, right? And the company for the longest time was like, but there's a Costco, you know, go to the Costco. And I was like, no, take all this stuff. You own the Costco. Take all those pieces, put them together in a meal, because that's what people want. They want the content component. So I think it's a cultural shift, and we've been making progress in that. I mean, just to even get to, you know, what I actually showed there, I mean, the beauty of it is, you know, unlike people that have been playing in this space, I don't have a whole lot to lose in the sense of I can be very aggressive in pricing. You know, you showed $7 a test. That's at list price for me. That's covering more drugs than would have been covered out there in the marketplace today, right? And people say, well, how do you make money on that? I've heard that from a couple people in this room. I was like, don't worry about that, is that we still can. The question is, does it actually resonate? If you have to put supplemental dollars in to get this price burden down, if really the question is you're trying to solve a problem, does that help you solve a problem? And I'm not trying to be all things to all people. You know, could we, you know, do we have networking capabilities on point of care instruments and even on the platform that I showed? Yes. Right? And if somebody's like, hey, we really want to tie into a group like a Thermo Fisher Scientific who has all this resources, we just need to be very prescriptive of what they actually need. We could be a powerhouse for, I think, a lot of other areas. It's just, it's new. The content piece is new.
So one interesting issue that always comes up, especially in the summer course that we do, is this movement towards decentralizing point of care, like the Cepheid box, right?
Yeah.
Or what Chandy was talking about, versus your interest in a more centralized lab platform. And people naively think it's either or, right? That is, if you decentralize, you have a gene expert Omni, there is no need for a centralized lab, we're going to shut it down. And in reality, we will need both, because you cannot do everything at the decentralized level, nor can you start MDR therapy at the decentralized level. So we will always need certain sophisticated capacity as a centralized lab level, but the market may be much smaller, because not every sample needs to be elevated to the centralized lab level. So how do you make a business case for a product that is really a niche product aimed at centralized laboratories?
Yeah. So I think it comes back down to is you want the answer of a next-gen sequencer at a price point of qPCR. How do you find that balance? And what you get from going from centralized to decentralized at the present moment, it's not to say, and I know, you know, as the technology, and I'll be the first one to acknowledge it, as technology is advanced, price will come down. But we always, I think we overestimate how quickly that clinical transition actually happens from a technology to the endpoint, especially in developing, you know, countries. I think it takes a lot longer. I mean, think about qPCR has been out there for 25 years, and still if you look at the primary technologies used in the marketplace, it's ELISA, right? Why? You know, I mean, it just highlights the fact people are like, oh, microarrays are totally going away. Now, you know, you can ask me later, you know, people say, well, why did Thermo Fisher just buy this archaic dinosaur of a company, right, called Affymetrix? It's microarrays. Well, it just kind of highlights the fact that these technologies are there longer than people realize. So when you look at that kind of content, you know, associated with it is how do you find a business case? You have trade-offs between the decentralized and the centralized component, and you have to at some point say what's good enough. Am I okay giving up on the content coverage and the decentralized model because I can only cover this much content at this price? Or do I need to move up a little bit further in the spectrum where if I go to next-gen sequencing, I'm going to get the best technical answer hands down, but it's going to cost me a lot, and people aren't going to understand how to do the bioinformatics and all the data. So then you can say, well, that's too high and this is too low. What's in the middle, right? So I think that's really what we're saying is don't be all things to all people, target, and if it's the right solution, it'll resonate and it'll make a difference.
Thanks a lot.
Yeah.
Michael,
Finally.
So if you look at the hype cycle for technologies, you've seen that famous graphic, right? Everything from 3D printers are hyped at one point, and people find a way to figure out where it fits into the ecosystem. I think sequencing is probably on the peak of its hype cycle, and you very nicely pointed out that it's not all easy, that there's a lot of translation challenges on sequencing. So do you think, say, in the 10-year horizon, we would come to a point when it will actually be of clinical utility in the countries that we care about? So if you take India, Chandy, you will know this. In all of India, 1.2 billion people, how many sequencers are there in India, you think? 5? Maybe 5, okay? So we really do not think of it as anything other than a research tool at this point in time. And even here, I think the clinical utility on a day-by-day basis is marginal at best. So what is your time horizon on when you think the product will reach a stage in pricing, in utility, the kind of challenges that you very nicely illustrated? Is it possible to overcome them? What is the time horizon you see?
I don't know. And I say that because the technology is still evolving. We haven't seen a plateau. So Illumina sequencing seems to be the dominant platform right now. But nanopore sequencing is just on the horizon. There's one company with a commercial product. There's several others coming out with new, more clever, more accurate ways of sequencing. It may completely invalidate or make a dinosaur out of the Illumina platform. I always come back, it depends what you want out of the diagnostic test, because there are sequencing platforms that can do very targeted, very simple types of assays, giving you very reliable, accurate answers. You can get that. I think what we're talking about is usually the comprehensive whole genome analysis. That is still in the on the horizon. But I would say this, again, I made the point about NIPD and then cancer diagnostics. That model works because they are currently in centralized facilities. So companies receive samples, they do the analysis, and they give back data. And in that model, it works. It wouldn't work where you had multiple sites trying to replicate the primary source.
Fabulous. We'll open it up to questions. Who had a question? Yes, Barry and then Ken.
Hi, I just wanted to ask Ellen, the model before was that rifampin was a marker for MDRs. And I think over time, and I'd be curious if you had data, we're seeing a lot of mono-INH resistant strains. Do you have a sense of what numbers or how much we're actually seeing that wouldn't be predictive of MDR if you just had rifampin?
It's very geographically dependent. And so I couldn't give you broad areas, but Panama, for example, it's 1%, and other places it's as much as 10%. So I think it varies within those ranges. But you couldn't give, it's individual parts of the world and individual parts of India, for example.
But you still made the decision to launch with just RIF, right? So not with RIF and INH.
This is, David Aland decided that RIF alone still had, I think it's mainly David's decision, Claudia may know better, but the secondary cartridge was a referral cartridge, and we talked about whether we wanted to add INH to the primary cartridge. But nobody was all that thrilled with the INH 2 markers either, and we didn't want to have a lesser predictive result.
And same with Chandy. You also only have RIF, right, Chandy, in your chip?
Yes, that's true.
Just RIF, okay.
But we have a follow-on test that's...
You have a follow-on test. Ken?
So, thank you. I really have enjoyed this whole session. So I'd like to play a bit of a devil's role advocate. starting with your observation, Madhu, about what transpires in India when you send trained patients. The reality goes back to 15 years ago. WHO Pierre Chollet, the late Pierre Chollet, had a practical approach to lung health, and they really demonstrated what I think drives this behavior, that for all persons who seek medical attention because of respiratory symptoms, only about 3 to 7% have TB. That means that 93 to 97% have something other than TB. And that's what drives that behavior. So as I look at the discussion of multiplex assay, I'm struck by the fact that we're still focusing on TB. I'm bundling the diagnosis with detection of drug resistance, but what we really need is a respiratory pathogens multiplex assay. And when you look at the literature, even the description of multiplex assay is almost genus or virus specific. So I'm going to look at viral respiratory pathogens in a multiplex assay, but I never look at TB. I never look for other bacteria. I think we really need to, so the question to the folks who are in the field of next generation sequencing, how far-fetched is it to aspire to have that sort of holy grail diagnostic capacity for any respiratory pathogen that people come in with, whether it's acute respiratory infection, whether it's flu, et cetera, but also not miss out on the diagnosis of tuberculosis.
Michael, you want to take that?
Yeah, I'll take that. So this is one of the strengths of next-generation sequencing, because you can do what is called a metagenomic analysis, where you can just isolate DNA from the lung and just sequence it. And then you can map those reads against large databases of known genomes to determine what's there, and in some cases, maybe even asking how much is there. So I think there, again, it's a discovery tool, because you don't know what the answer is, and you're asking the question, what's there? That's when it's a very powerful technology.
Go ahead.
Oh. Thanks. I'd like to ask a question on behalf of the children who are suspected of having TB. And being a pediatrician, looking at these 500,000 to 1 million cases of childhood TB that are estimated each year, and seeing that diagnostic methods that are looking into detection of the actual pathogen in respiratory or related samples in children don't seem to perform very well, with estimates ranging around 30 or to 60% of childhood TB cases confirmed by such technologies. Is there any approach that looks at alternatives? I mean, childhood TB diagnostics has been using methods that have first been established in adults and seeing suboptimal results in many. So is there an alternative pathway that we can look at?
I guess nobody on the panel has really put in much work on TB, but the expert does pick up a lot more TB in children.
As we have discussed before, they're using it in urine, and I think
Gastric
children
juice
probably have more circulating TB in urine, just like HIV patients, because they have diffuse lung disease. They're using it. We've worked with stool, but Andrew DiNardo and I have a publication about using the string test for children and pulling it out and running it on the Gene Expert. It was very sensitive in that situation, and it certainly beats gastric aspirate. So I think a more sensitive test can be used on alternate samples than a respiratory type sample if the organism is present. And I think Steve and Lon's papers were fairly surprising about how many people had it in their urine. So I think more studies need to be done. But I agree with you, if we had biomarkers that you could use from blood or something better, then we would certainly go to that kind of a test.
Yeah, and I would just add, we have a similar type of program that's more on the antibiotic resistance, which is outside of TB, which goes to kind of the respiratory components and looking at broader coverage. And we started actually with fecal samples as well as urine. So we've proven that it's actually working through some of our collaborators. But I think to your point, one of the initial things that comes out is in pediatrics. So it's an area that we're interested in, but I don't have any specifics for you at the moment.
So we are out of time, and we have to close, but I want to give Claudia an opportunity to close out the session with her thoughts, but I wanted to thank each and every one of you for your candid and honest responses. Thank you.
Thanks very much, Madhu, and thanks to the panelists. I think this was a great overview. Deborah is already hovering. Food is out there waiting. So thank you very much, and we look forward to the afternoon.